I'll just talk in a few minutes about our laboratory in Egypt we established in 2014, Cancer Nanotechnology Laboratory. We are working with different types of tumor-targeted drug delivery nanomedicine, including passive targeting, active targeting. Also, some work we did about cancer nanotheranostics for imaging breast cancer and liver cancer in addition to therapy. And finally, we developed some inhalable nanocomposites for lung cancer targeting. We have made some patent applications of this, and we have some few publications about this in high journals of drug delivery. That was our lab in Egypt. Then I moved to US, where I am a postdoc now in Harvard Medical School. So I'll talk a little about cancer immunotherapy. Of course, Dr. Hazem covered all the aspects of epigenetic reprogramming and how to enhance cancer immunotherapy, but I'll talk from an engineering point of view or drug delivery by nanomedicine. So all of us, I'll not talk about the introduction of immunotherapy and how T cells attack the cancer cells, because Dr. Hazem already covered very well. So this is just the basic steps for effective anti-tumor T cell response. When some antigens of cancer cells released, it is taken by antigen-presenting cells or dendritic cells, which will induce priming and activation of T cells. After that, T cells have to infiltrate the tumor microenvironment, where it interacts with the cancer cells and recognition of the antigen of the cancer cells, then killing the cancer cells by different mechanisms like perforin, organ release, or different other mechanisms. And like Dr. Hazem said also, T cells have many accelerators, or we can say activating signals, in addition also to inhibitory signals. Some of the most important inhibitory signals like the PD-1 and CTLA-4 antigens overexpressed on the T cell. So how the immune checkpoint inhibitors work, I'll not talk much about this. We all know the interaction between the overexpressed CTLA-4 on the T cells with the complementary protein on the dendritic cell. This interaction prevents dendritic cells from priming T cells to recognize tumors. So they developed immune checkpoint inhibitor called ipilimumab. It's antagonist or antibody to this protein to block this interaction, then reactivate again the T cells. Also the PD-1 inhibitors, the same concept, but it blocks the interaction between PD-1 on the T cells and PD-L1 overexpressed on the cancer cell. That's how the immune checkpoint inhibitor work. But like we know that large proportion of patients still not responding to the classical immune checkpoint inhibitor. And there are many reasons for development of resistance to immune checkpoint inhibitor. One of the reasons, the epigenetic modulation, like Dr. Hazem explained. I'll talk about different other reasons like low immunogenicity of cancer cells, for example, and impaired antigen presenting capacity of dendritic cells, and also the activity of CD8 positive cytotoxic T lymphocytes can be suppressed by different types of other immunosuppressive cells in tumor microenvironment. We cannot neglect like tumor-associated macrophages, myeloid-derived suppressor cells, and T regulatory cells. So this is just a figure summarize the reasons of resistance development to immune checkpoint inhibitor. It can be like downregulation of antigen presentation, like here. So it will lead to loss of signal 1 that come by interaction of major histocompatibility complex and T cell receptor. It can be reduced the expression of some co-stimulatory ligands. It lead to loss of signal 2. It can be also by interaction of PD-1 and PD-L1, or the other immunosuppressive cytokines and growth factor like TGF-beta that we said now, and other factor like interleukin-10 and VEGF that reduce the activity and make exhaustion of T cells. So how to overcome those challenges? Researchers think about novel tactics that can be explored for reprogramming of the immunosuppressive tumor microenvironment. So now the fight or the battle is not only against the cancer cells, it's actually against other immunosuppressive cells in the tumor microenvironment, how to reverse this immunosuppressive function in order to reverse or reactivate the T cells again. So I'll just talk in very briefly about some techniques to enhance those or the activity of those suppressed T cells. Number one, some researchers engineered nanomedicine for induction of immunogenic cell deaths. What is meant by immunogenic cell deaths? The idea is to just enhance the release of some molecules called damage-associated molecular patterns or DAMPs from cancer cells, like calreticulin or HMGB1 or ATP or interferon. Those molecules can activate the dendritic cells or antigen-presenting cells, like you see here. So after treatment of cancer cells with some immunogenic cell inducers like doxorubicin, like oxaliplatin, or some other drugs, it enhances release of these molecules of DAMPs. These molecules can induce recruitment and the maturation of dendritic cells, so that dendritic cells can enhance its antigen-presenting capacity and the priming of T cells. T cells become able again to attack the cancer cells and kill it. So in our laboratory here, we are developed, but this is unpublished data yet. This is type of multi-compartmental nanocomposites. I'll try to explain or simplify the idea of this engineered system. This is simply liposomes containing some drugs that act by inhibition of some mutations in melanoma cells, like BRAF, for example, and also MEK inhibitors. So we have encapsulated those drugs, BRAF inhibitor and the MEK inhibitor, into a liposome by certain method of conjugation to some of the phospholipids incorporated in liposomes. Then we have encapsulated into the aqueous core of liposomes some other nanomedicine, very tiny, about 40 nanometer. It is composed of lactoferrin conjugated to the doxorubicin. Doxorubicin in low doses can induce immunogenic cell death. Then we have modified the surface of these liposomes with anti-PD-L1 monoclonal antibody. This antibody here acts as both targeting ligand to cancer cells that can overexpress this protein, and also it helps in internalization of liposomes into cancer cells to release those drugs that inhibit the mutated enzyme BRAF and also it will block the interaction between PD-1 and PD-L1, resulting in activation of T cells. So this is just for if someone interested in the chemistry of conjugation, how we conjugate the lactoferrin molecule. Lactoferrin is a macromolecular protein or glycoprotein that have some amino groups. We worked on this amino group to conjugate with hydrazinobenzoic acid, but we used FMOC protected hydrazinobenzoic acid to prevent the intra-cross-linking of lactoferrin. Then carbodiimide reaction between the carboxylic group of the hydrazinobenzoic acid and the amino group of our protein, lactoferrin, have been established. Then we added the doxorubicin here as an immunogenic cell-inducing agent to generate hydrazone bond. The hydrazone bond is a type of pH-responsive bond that can be cleaved only on tumor cells. So the unique character about these nanoparticles that it retained the same structure and the same molecular size of lactoferrin without any aggregation or intra-cross-linking, about 40 nanometer. Actually, we need this size below 50 nanometer nanoparticle because it shows very high penetration ability into tumor and can reach the tumor cells in the core of the tumor that is usually responsible for resistance. And also it has high drug loading of the drug. So we have very small nanoparticles of lactoferrin doxorubicin for induction of immunogenic cell death. And we proved the conjugation by different techniques like NMR spectroscopy. We have also measured the size and surface charge of the nanoparticle and the internalization into cancer cells and cell viability. Then we encapsulated those into another type of liposomes. I mentioned before that encapsulated the BRAF and MEK inhibitors, and we have characteristics. So now the system is like liposomes encapsulating the BRAF and MEK inhibitor and encapsulating into the aqueous core the immunogenic cell-inducing tiny nanoparticles of doxorubicin lactoferrin. We have evaluated the release and everything. Also the binding of our liposomes to cancer cells. Based on the PD-L1, we have optimized the amount of PD-L1 monoclonal antibody on the surface of nanoparticle based on how it induces the binding to cancer cells that should express this protein. And we pre-treated actually the cancer cell with interferon gamma to increase the expression more of PD-L1 on these cells. Then we measured by flow cytometry how the binding is affected by the amount of PD-L1, and it was clear that by increasing the amount of PD-L1 on the surface, antibody I mean, on the surface of the liposomes, it increased the binding to the cancer cell. We have evaluated also the cell viability onto different cancer cells and also on fibroblasts to see the targeting. Here also we made immunofluorescence staining to prove how our anti-PD-L1 targeting technology increased the internalization of liposomes. You can see here liposomes containing or encapsulating a fluorescent dye, that is FITC in green color, how its entry or internalization into the liposomes have been maximized compared to the non-targeted liposome, using here the red phalloidin as a counter dye. Then we have evaluated how our nanoparticle-induced immunogenic cell death by measuring different factors. I can mention here the calreticulin exposure and how treatment of cells, cancer cells with our nanoparticle increase the exposure of calreticulin compared to the control or of the free drugs. Also, we have evaluated this exposure of calreticulin, one of the immunogenic cell death molecules or indicators by flow cytometry to see the early and late apoptosis. I'm sorry not to share more of the data here because it's still unpublished, but I'll just shed light on different other nanomedicines that can help or enhance the efficacy of immunotherapy by different ways. To see that the battle now is not only against the cancer cell, also against different other immunosuppressive cells in tumor microenvironment. Let's take this example of TAMs or tumor-associated macrophages. This is one of the cells present in the tumor microenvironment. Actually, it has two phenotypes, M1. M1 has anti-tumoral effect or response and can phagocytose the cancer cells actually and degrade it. However, M2 TAMS has the opposite, has pro-tumorigenic activity, and the cancer cells can induce polarization of TAMS from M1 to M2. M2 has immunosuppressive function. It cannot induce phagocytosis of cancer cell. On the contrary, it enhances the angiogenesis and the metastasis more. So we wanted to increase the polarization of TAMS present in tumor microenvironment to M1 macrophages rather than M2 macrophages. So to do that, let's have a look on how phagocytosis occurs. There is some pro-phagocytosis, or we call it eat-me signals. But this due to interaction between some molecules on the surface of tumor cell, like the normal antigen, tumor-associated antigen, or other molecule like the calreticulin that we talked about now, with some complementary receptors on the phagocyte or macrophages. Due to this interaction, complementary interaction between cancer cell and phagocyte, this could induce tumor phagocytosis, and the macrophage, M1 I mean, can phagocytose cancer cells by this pathway. However, we have also balance with another anti-phagocytosis, or we call it don't eat me signals between cancer cells and the phagocytes. This also can be mediated due to interaction between some factors like PD-L1 on the cancer cell or CD47, this is very important one, and their complementary proteins on the phagocyte. If this interaction occurs, this will inhibit tumor cell phagocytosis. And of course, cancer cells trying to increase the exposure or upregulate these factors to make the balance to the side of anti-phagocytosis so that it can survive and overcome the function of macrophages. So in our laboratory, they developed some idea that can enhance phagocytosis activating nanoparticle. How this? Like you see, this is the M2 macrophage that has pro-tumorigenic effect. Actually, cancer cell induce polarization of the macrophage to the M2 type by release of factor called colony-stimulating factors that can bind with complementary receptor on macrophage and induce M2 polarization. At the same time, like we mentioned in the previous slide, cancer cell overexpress CD47. This CD47 is a don't eat me signal, anti-phagocytosis signal. When it binds with SIRP alpha on the macrophage, it inhibits the phagocytosis. So here, this is a type of nanoparticle that is targeted or surface modified with antibody against that receptor, SIRP alpha receptor, to block the interaction between CD47 and SIRP alpha, so that we inhibit this anti-phagocytosis pathway and induce phagocytosis of cancer cell by macrophages. At the same time, this drug, AK750, is a patented drug. This drug is blocker for the this receptor, colony-stimulating factor receptor. By blocking this receptor, actually, it induces repolarization of M2 macrophage to M1 that can induce phagocytosis of cancer cell. This is some of the results. The nanoparticle could reduce significantly the growth of tumor by this mechanism, and also by quantification of immunophenotyping, I mean of the tumor microenvironment, they found the M1 macrophage percent increase rather than the M2 that can ensure the successful polarization of M2 to M1 macrophages. This is some of the images that show how macrophages can phagocytose cancer cell. Maybe this is more clear images. These macrophages in green color can phagocytose and degrade the cancer cells that is labeled red by cherry red. Another type of, or another approach of how to make the nanomedicine reprogram the tumor microenvironment to enhance the efficacy of cancer immunotherapy by metabolic remodeling. Of course, the cancer cells utilize high amounts of glucose by aerobic glycolysis, or what is called Warburg effect, to produce energy and produce a lot of lactic acid or lactate. Actually, the shortage or glucose-deprived lactic acid enrichment tumor microenvironment can impair the effect and proliferation and function of T cells. So this will result in reduced response to immune checkpoint inhibitor. There are another mechanisms of different metabolites, like, for example, also the competition between cancer cells and immune cells on amino acids like arginine. Cancer cells can overexpress the arginase 1 enzyme that can metabolize arginine. So by decreasing the level of arginine in tumor microenvironment, this also will affect and will inhibit the proliferation and activation of T cells. So if we can inhibit this enzyme by a drug like arginase inhibitor, this can maintain the arginine level in tumor microenvironment and allow again of reactivation of T cells. Also, there is enzyme indolamine dioxygenase. This enzyme is responsible for metabolism of tryptophan into kynurenine. That is also cancer cells utilize this pathway of metabolism. And the byproduct, kynurenine, has inhibitory function of the T cell activation. That's why if we inhibited this enzyme by drugs like indoximod or ipacadostat, these drugs can inhibit the formation of kynurenine and reduce its inhibitory function of T cells. This is just examples of how by regulating the metabolic pathways, we can reactivate the T cells again. Also, there is a very important pathway of metabolism, glutamine metabolism. Glutaminase enzyme can hydrolyze or catabolize the glutamine into alpha ketoglutarate. Alpha ketoglutarate is a metabolite that is known to promote M2 macrophage polarization. And we said like M2 has immunosuppressive function. So by inhibiting this enzyme with those drugs, it can reactivate the macrophage of macrophages from M2 to M1 that has anti-tumoral effect. This is a type of nanoparticles published that has this drug is glutaminase inhibitor combined with another metabolic drug is metformin. This inhibitor of glutaminase enzyme encapsulated into polylactic glycolic nanoparticles, it's found it reduced the tumor growth very significantly compared to the control. Here, I will not talk about, of course, the details of epigenetic reprogramming because this is very well covered by Professor Dr. Hazem. We know now that by epigenetic modification, we can convert the immune cold to immune hot tumor microenvironment. And by epigenetic agent, we can decrease the abundance of TAMs, of course, the M2 TAMs and the myeloid-derived suppressor cells, and increase the infiltration of cytotoxic T cells. For example, hypomethylation agent that we know now, like DNA methyltransferase inhibitor, for example, drug called zebularine. I just talk very briefly. Zebularine is a DNA methyltransferase inhibitor that induce hypomethylation, like Dr. Hazem explained. If we combined with PD-1 antibody in the same nanoparticle, this can be very effective therapy and increase the effect of PD-1. And here the nanoparticles act as a targeting agent so that we can make more selective targeting to certain type of cells rather than other cells in the tumor microenvironment. Finally, there are now new approaches of local cell-based immunotherapy. For example, some researchers encapsulated activated dendritic cells into alginate microspheres and used these microspheres as like vaccinations mode. By injecting subcutaneously, it will activate the immune system because it's already containing. So here the trend is to encapsulate the immune cells, activated immune cells, and introduce or inject into the animal to induce more powerful immunogenic anti-tumor response. There are also, not to take so much time, I know time is limited, another publication that encapsulated tumor-specific T cells after some genetic engineering of the T cell receptor into implantable alginate gel. This implantable alginate gel contains these activated T cells and contain also mesoporous silicon microparticle that has some antibodies like anti-CD3 and CD28 antibodies. Those are agonist, not antagonist antibody to activate the T cells. Then injected into the tumor, intratumoral, that very simple injection. This is the alginate hydrogel. Drug delivery scientists know how to generate this type of hydrogel, then injected into the tumor to induce immunogenic response. And finally, this one of the projects I'm working in, but unfortunately still unpublished, so I can't disclose some. We are trying to micro-encapsulate some of the T and B cells into microparticles prepared by a technique called microfluidic technique that produce very uniform microparticle with the same uniformity, the same size, and very mild conditions that not affect the activity of immune cell. Some old researchers talk about microencapsulation of different cells by using this technique, but no one encapsulated like those immune cells and maintained this activity till now. I hope it can be published soon, then we can discuss more. Thank you so much.